Hi guys, microscopes part 2. Last time we learned what the parts are. Today we're going to learn a little bit about what some of the parts do and how to get the thing up and running so you can actually look at stuff. Okay, so we talked about this being the body tube and the eyepiece that you look through. Okay, and we also talked about the coarse adjustment knob and that these are your objective lenses. You have 3 objective lenses. You have your low objective lens, which is the shortest one, and it has a red color on it. Your medium objective lens magnifies even further. It is yellow and a little bit longer. And then the longest one is your high objective lens, and it is color coded with a blue. Low objective magnifies 4 times. Medium magnifies 10 times, and high magnifies 40 times. Okay, you always want to start on the low objective lens because it's the easiest one to focus on. Okay, you have stage clips, which I said hold on your sample. Your sample is going to be on something called a slide, which is simply a piece of glass, and you guys actually use these when we did scratch tests for rocks and minerals. This is where you put whatever it is you're looking at on the microscope. It goes on top of this slide. And then we have something called a cover slip, which is either a small piece of plastic or a small piece of glass, and it holds down the specimen so that it doesn't fall off or crawl off, depending upon what you're looking at, of your microscope slide. Okay? Once your slide is prepared with the specimen, you put it on the stage and you hold it over the aperture. The aperture is the hole in the middle of the stage. And then you use the thumb clips or the stage clips with your thumb to hold it down. Remember, you start with your low objective lens. You'll hear it click into place. And you drop the body tube using your coarse adjustment knob all the way to the bottom. Okay, that's going to be pretty close to focus there. And then what you'll do is you'll look through with one eye and slowly raise the body tube until your object comes into view. Once your object comes into view, you will use the smaller knob, the fine adjustment knob, to get the blurriness out. This will fine tune it and make your image much sharper to look at and easier to see. Now remember one thing, this is a trick. When you decide you want to increase your magnification and move to your medium objective lens, you should already be in the right position to focus and should simply have to rotate the nose piece until the medium objective lens clips into place. Once it does, it should already be in focus or very, very close to focus. So from here, you will just use your fine tune adjustment to make your image sharper. Okay? And likewise, when I really want to blow it up, I simply need to move the body tube into a high adjustment position, and it should be in focus. I might tweak it a little bit using the fine adjustment knob, the small knob, but I should be really close just by moving the body tube. Okay? You look at whatever you want to look at, and some things are better looked at using the low objective. Some things are better looked at in the medium. Some things are so tiny you won't be able to see them very well until you look at them with your high objective lens. But also, some things are going to be a little too big. Your high objective lens won't really show you much. It'll blow it up too big. Okay? Now, when I'm done looking at the specimen, I want to change slides. I always change back to my low objective position, and I raise my body tube to the top. Okay? This gives me plenty of room to maneuver. This makes sure that when I move my slide, I don't bang into the objective lens. If I bang into it, I could scratch it. So this gives me plenty of room to change slides, change specimens, and then put something new on to look at, clip it in, and then again, drop it all the way to the bottom and start that process all over again. Okay? Power magnification is the magnification of your eyepiece times the magnification of each objective lens. Okay? Today, what I'm going to have you do, and I will post this in Google Classroom, there's a link below that takes you to a virtual microscope lab. You will have to prepare your slides. You will have to put a cover slip on it. You will have to choose between a human cell, a different type of animal cell, or a plant cell. You will prepare the slide, put on the cover slip, and you will have to focus in low, medium, and high objective. You will also have to read through some of the ways that we calculate the magnification using math to calculate that using the eyepiece magnification, not the eyepiece magnification, the eyepiece magnification and the magnification of each objective lens. Okay? So you'll read through that and then you will play around with the microscope. Follow the directions on the website. It's virtual. Click and drag. You'll get to blow up some images of some different types of cells, and it will give you some information for you to be able to fill out also the attached worksheet, which you can absolutely type into. Okay? Don't forget to turn it in when you're finished with it. This assignment will be due on Monday. Okay? So you'll get it this morning, which will be Thursday when you see this. And this assignment is due on Monday. Okay? And there will also be, actually, there will not be a video meeting on Monday. Okay? Or, yes, on Monday. Okay? That's a day off, so take a day off. But we will have another video meeting again on Thursday. Okay? 12 to 2 is the set time for us. And you can ask me questions. You can always reach out via email as well if you want to, or a comment on Google Classroom. Okay? See you soon, guys. Take this seriously. Get it done. Have a great day. Bye.